Good morning, everybody. I am Muhsin Akdoğan. Today I will be presenting a story from a real story from a CDV, Canine Distemper virus, infects dogs and is a fatal disease. Here I would like to put an introduction and mention about the goal of my study. The canine distemper virus is a fatal virus, is a single RNA stranded, single molecule stranded RNA virus, and is fatal. And as you can see from the title, I put a colored, the text are colored. It's a multisystemic disease that causes effects on respiratory system, digestive system, respiratory system, and has a lot of effects. Here the contents are following with my Galaxy history and the data sets that I used, the NGS analysis methods that I applied, and the results following with the, I will be mentioned in more detail in the following. And it was a really intensive research for me. Here the data sets that I used. Here, as you can see, there are four. I have used 12 different samples, each around 5 gigabytes, in total like 60 gigabytes. 4 mock data, 4 OND, which are attenuated viruses, and for the R252 virus strains that are the causes the disease. To continue with the NGS analysis methods that I applied is first of all I have reached the raw data and apply FastP to investigate how this data is concise, and apply multi-QC to see overall 12 sample success, and later apply the mapping and alignment with RNA star, and later apply again multi-QC to see how alignment is successful here, and continue with the feature counting to select the most aligned reads to RNA star results and apply DSEC2. And later, before doing enrichment and analysis, I applied filter data sets with 2 different parameters that I will be talking about them. Here I will skip the data processing and maybe talk here about the quality. Here, as we can see, there are the GC content, the fresh scores, and the reads after filtering results. As well, in the right side, we observe the insert size distributions with 2 peaks. That shows us the read lengths are not random here, but have some patterns. As well, we observe the past filters with the blue size that are really significant, and our data is good. And below we observe the GC content about 55%. And in the right side, the sequence quality, the FRET score is above 35, and you don't see because of this machine, but there are here 12 different samples. And the read and content, the mismatched reads and non-reads are again good. Here there are the RNA star results, the aligned and unique aligned above 70% scores. All of them. This data is similar to, but in more detail to them. Similarly, that one shows the uniquely mapped and unmapped reads. We can observe the uniquely mapped reads are present in a significant amount here. And later, after I observed my RNA star results are good in my multi-QC analysis, I go and divide those results. At the beginning, I set up to all samples, but later to 3 samples, R252 mock and OND, the attenuated viruses, to further make feature counting and filtering on them before arrangement and differential gene expression analysis. Here I do have three different groups. The MOC R52. Here is an away plot that we observe the upregulated and downregulated gene here, and we do observe they significantly distributed from each other in MA plot, and the dispersion estimates the black, blue, and red colors that show this pattern. But here in principal component analysis, we have to we should expect to see that those results are clustered to each other than that. Before I conducted that study, I have started with only 6 samples and conduct all my study with 6 samples. But in that study, there are 12 samples because I rerun the study. I observed those results not enough to present my study, and I increased my sample size to 12 to eliminate those things in my research. Because when I finished my study with 6 samples and do enrichment analysis, I left with only few genes that cannot support my study for that. But when I increased to 12 samples, as you can see at the end, there are a lot of differences. Here, similarly, the OND and MOC results has a similar distribution in terms of MA plot, but when we compare these 3 MA plots, we can observe at the OND versus MOC results, there is more intensive data and more abundant data here that we'll talk about at the end about those results. Here, the filter data sets I have set to 5%. I have filtered my data with 5% that are the most significantly upregulated or downregulated. And in the second parameter, the third column, the log fold change, I have said that I want at least 2-fold increased or decreased differential gene expression and filtered out my data. At the beginning, there are like dozens of genes, and later I left with 1,000 for each my group. Here, as you can see, before filtering, this is filtered with 5%, those with 5%, and those results with both 5% and the log fold change above the one. And there is not much differences, but we observe a reduced number. And finally, before going to enrichment and analysis, I have put my data to a bioinformatics tool that I find in the University of Ghent bioinformatics website, and created a Venn diagram where I put 3 different groups: the OND, R2, the OND and disease-causing agent, the disease-causing agent and the healthy one, and the OND and the healthy one, and observe those distributions. And later I go to Go Profiler and observe what are those causative in each of them in the disease. Here in biological interpretation and discussion, I can say that here we do observe 713 different genes that belong to OND expression. OND is the attenuated virus, and this is the disease-causing virus in R252. When we look to 713 different genes, that shows the success of the vaccination here in the dogs. And when we look to the enrichment analysis and the genetic profiles, metabolic pathways in Go Profiler in terms of cell components, metabolic functions, phenotypic pathways and metabolic pathways, I observe those in 713 in attenuated viruses. There are increase in the cytokine signaling, This cytokine signaling is started up onto immune system in the innate and adaptive system. Cytokines triggers those immune responses, and they are upregulated when the OND in the OND samples, which is suppressed in the disease, and that shows how those vaccine strategies are effective in upregulating the genes that are suppressed with the disease formation. And here, before ending my presentation, I will be briefly talk about, as I mentioned, the metabolic functions, the biological process, cell components in the red, orange, green, and purple, the KEGG, Kyoto Encyclopedia for Gene and Genome, and this HB for the phenotypes. Those are enriched with the cell surface and cytoplasm related cell components like tight junctions. The viruses weakens the tight junctions to further invade other cells. But in summary, when we can observe, we can comment specifically in each of them what they cause. But in summary, these 713 genes in general has role in extracellular region, cell surface, positive regulation of cytokine production, cellular response to stimulus chemical, CAMP response, which are the messengers in the cell signaling, and catalytic activity. As well, we also observe in cell cycle related molecules that are affected here. And here I try to briefly mention about those causative genes, and when I would like to talk about the references, how I have used my references here. This one, the data sets that I used, the 12 different sample, and
I think you don't need to go with the reference.
Tamam hocam. That's the end of my presentation. Thank you for your attention.